Right, so now we're going to take a look at some viruses that are going to infect animal hosts. So we just looked at all of the bacteriophages and went through the lytic process and the lysogenic process, and then even looked at transduction. And now we're going to look at viruses that are infecting animal hosts. So lytic animal viruses follow similar infection stages to the bacteriophages. That's why we looked at bacteriophages first, so we could look through the process in a simple cell, and now we're going to talk about it in a little bit more detail in a complex cell, which would be like an animal cell. But it goes through the same stages. We have attachment of the virus to the cell, penetration where it's getting into the cell, it's a little different here, but biosynthesis, then maturation, and release. However, the mechanisms of penetration, nucleic acid biosynthesis, and release differ between the bacterial and animal viruses. So this is where we see a difference. So after binding to the host receptors, the animal viruses are going to enter through endocytosis. So I'm going to see if I can scroll down here so we can look at the words at the same time. Here we go, at the same time as the illustration. Which remember, endocytosis is engulfment by the host cell. So it's bringing in something into the host cell. So we see the same or a very similar type of binding to the cell. So we have our little proteins on the outside of an animal cell, for example, and then we see our virus is going to attach itself to these little proteins, and then that's going to start it going through this process of infecting the cell. So it's going to bind to these host receptors, and then the animal virus is going to go through endocytosis or through membrane fusion. Okay, so endocytosis is engulfment of the host, or engulfment of the bacteria, or engulfment of the virus by the animal cell, by the eukaryotic cell. And membrane fusion is when that viral envelope, if it has an envelope, is going to fuse with the host cell membrane. So they're going to just meld with each other, and we'll see that in just a moment. So again, many viruses are host-specific, meaning that they only infect a certain type of host. So only human, for example, or only bird, or only swine. But most viruses are only infect certain types of cells within tissues, and we talked about this at the beginning of the chapter. But this type of specificity is called tissue tropism. So it's when a certain virus only infects a certain type of cell within a certain type of organism. So examples that they give here are the poliovirus, which has tropism for tissues of the brain and the spinal cord, very specific, or the influenza virus, which has tropism for the respiratory tract. Okay. So let's take a look at the image here, the illustrations. So we have our attachment phase, right? So we have our virus, this example is using influenza virus, is going to become attached to the target cell. So we have the little proteins in here, and then they're going to be attached to the cell. And then penetration, in this case, when we're not talking about bacteriophages, but we're talking about eukaryotic cells or animal cells, then what we see is the cell is going to engulf the virus. So you can see right here we have an invagination, if you remember that term. An invagination, this is where the cell membrane is dipping down, and it's going to pull up and around that virus. So then what we see is it engulfs it, and it's going to bring it into itself. And you can see here that it even includes some of the membrane, because as it engulfed it, it has the membrane around it. Then we have what's called encoding. So this is where our viral contents are going to be released into the cell. So it's going to burst through that membrane that it has after it got into the cell, and then we're going to release those pieces of the virus into the cell. Then we go through biosynthesis, so similar steps to our bacteriophage, biosynthesis. In this case, viral RNA is going to enter the nucleus. So you can see, remember, in this one we have a nucleus, so we have a viral RNA is going to go through a nuclear pore, get into the nucleus, and then it's going to be replicated by the viral RNA polymerase. So viral meaning the virus brought it with it. Remember, it has all of these necessary components inside of its capsid. It's going to be replicated by the viral RNA polymerase. Now, this is just an example. We're going to talk about some different types in just a moment. But in this case, we're saying that this virus brought RNA. That RNA gets into the nucleus, and then it uses viral RNA polymerase to replicate itself. Here you can see it has replicated itself. There are multiple copies here. Then it goes through the step of assembly. So new phage particles are then assembled. Okay, so our RNA is going to act just like mRNA in a typical eukaryotic cell where it makes proteins. So we have transcription, translation. So then it translates it into proteins that are needed to make new viruses. Then we have our new viral particles are made and then released into the extracellular fluid. So then when they're in the extracellular fluid, then the cell, which is not killed in the process, continues to make new virus. And we're going to take a little bit closer of a look at that in just a moment. So let's look at the words here. So if we go back up in the words where we left off, was that we talked about it only infects a certain type of cell. And we got to the point right here, after it binds to host receptors, the animal virus is going to enter through endocytosis. So one, it's going to engulf it and bring it in, so it'll have membrane around it, or through membrane fusion, which is where the membranes will fuse, given the term. So the viral envelope is going to fuse with the host membrane. And then what will enter the cell will be the capsid, and then the stuff inside of the capsid. So animal viruses do not always express their genes using the normal flow of genetic information. So this is where we're going to talk about some of the differences. So once it gets into the cell, either by endocytosis or fusion, then we're going to see some differences here based on the genetic information that the virus is bringing into the cell. So we can have different types of information based on the DNA, if it's RNA, and then that process going from DNA to RNA to protein may be different based on what's brought into the cell. So some viruses, as we have already mentioned previously in the chapter, have double-stranded DNA, dsDNA. So if it's a double-stranded DNA genome, that's just like the cellular organism. So just like the eukaryotic organism has double-stranded DNA, just like all of our cells do, and then it can follow that normal flow. So if it has double-stranded DNA, it can integrate itself into the host cell, or it can just use the cellular components, the different mechanisms, the machinery in there, and then simply use what's already in there to make RNA, to go through translation to make RNA, and then transcription to make proteins to make copies of itself. However, not all viruses have double-stranded DNA. Others might have single-stranded DNA, ssDNA. They might have double-stranded RNA, or single-stranded RNA genomes. And so then we see a difference here. The nature of the genome determines how the genome is replicated and expressed as viral proteins. So if a genome is single-stranded DNA, so we already talked about double-stranded DNA. It works just like double-stranded DNA in all of our normal cells. If we have single-stranded DNA, then the host's enzyme, so once the virus infects a cell, it's going to use the host enzymes to synthesize a second strand that is complementary to the genome strand that it has. And then it will get to that double-stranded DNA point, and then the double-stranded DNA can now be replicated, transcribed, and translated similar to the host DNA. So if the virus infects a, let's say, human cell, once it infects it, if it's double-stranded DNA, then it just goes through normal cellular processes. It's going to use all of the machinery inside of one of our cells to make copies of itself. If it's single-stranded DNA, then it will use the machinery inside of the human cell to make the complementary strand. Then it has double-stranded DNA, and it can go through the regular process of transcription and translation. If the viral genome is RNA, then we have a completely different mechanism that is used. So there are 3 different types of RNA genomes. There is double-stranded RNA, there's positive single-stranded RNA, or negative single-stranded RNA. So if a virus has a positive single-stranded RNA genome, then it can be translated directly to make viral proteins. So our positive single-stranded RNA acts just like cellular mRNA. So remembering back to cells, our eukaryotic organism is going to make mRNA, and then that mRNA is going to be translated into a protein. If, on the other hand, it has the negative single-stranded RNA genome, then we can't just directly go into making mRNA, or we can't directly go into translation because it's not like the mRNA in a eukaryotic cell. It's not like the positive single-strand RNA. The positive stands for the sense strand, which is the strand that's used to make proteins. The negative stands for the antisense, which is the opposite strand. And if it's the opposite strand, that is not the strand that is used to make proteins. So then what needs to happen is that we have to take this antisense strand, or as it's labeled here, the negative single-stranded RNA, and we need to replicate it into a positive strand. And the way that the virus does this is by using viral RNA-dependent RNA polymerase, or RDRP. So this RDRP is brought in by the virus, as it's mentioned, so it's viral RNA-dependent RNA polymerase. So the virus itself brings this into the cell. So it uses this enzyme then to make the positive strand, the positive single-strand RNA, from the original negative single-strand RNA genome. This RDRP, and then since it makes the positive single-strand RNA, then it would be just like this mRNA, where then it's able to go on and go through translation in order to make the proteins necessary for it to replicate itself. The RDRP is also an important enzyme for the replication of double-stranded RNA viruses. It uses the negative strand of the double-stranded RNA genome as a template to create another positive single-strand RNA. So then in this case, we have the positive strand of RNA, and then we have the negative single-strand RNA, and then the RDRP is used to make an additional single strand that's positive of RNA. So then this newly synthesized positive single-strand RNA copies can then be translated by the cellular ribosomes and then make more proteins. So we see RDRP being used when the genome is a negative single-strand RNA, but we also see RDRP being used when it's a double-stranded RNA. And we can kind of see that in an illustration here. So RDRP, again, is viral RNA-dependent RNA polymerase. If we have a positive single-strand RNA, then we can just go directly into making viral proteins using the host ribosomes. If we have the negative single-strand RNA, then we have to use RDRP in order to make the positive single-strand RNA to then make our viral proteins. An alternative mechanism for viral nucleic acid synthesis is observed in retroviruses. So we have an additional way or an additional set of roles that we see for what's called a retrovirus. So retroviruses are positive single-strand RNA viruses. The single-stranded RNA viruses such as HIV carry a special enzyme called reverse transcriptase. So HIV is the main one that people talk about when they talk about retroviruses, so we'll use that as an example. So HIV is what's called a retrovirus. It contains the positive single-strand RNA, and it also, within it, contains this reverse transcriptase, which is a special enzyme. And it's within the capsid, and that synthesizes a complementary single-strand DNA. So it's reverse, as the name implies. So typically what we see is we have RNA, or rather we have DNA, and then from DNA we make RNA, and then from RNA we make our protein. What we see in a retrovirus then is that we start out with our positive single-strand RNA, and then using reverse transcriptase we are going to reverse transcribe and then make our single-stranded DNA, what's called cDNA. So you can see it's actually kind of going backwards in this the way that we normally see things work, it's going backwards. So it's reversing, it's reverse transcribing for reverse transcriptase. So it uses this positive single-strand RNA genome as a template, and then it makes the single-stranded DNA, and then the single-stranded DNA is then made into a double-stranded DNA. And then that can integrate into the host's chromosome and then become a permanent part of the host. So since we are able to take this single-stranded RNA, make a single-stranded DNA, which then goes on to make a double-stranded DNA, then it's just like the host genome. It's just like all of our cells that contain double-stranded DNA. And then it integrates itself into our chromosome. So the integrated viral genome is then called a provirus. A provirus, just like we saw a prophage earlier when we were talking about phages and bacteriophages. So the virus can now remain in the host for a long time. And this is how we establish a chronic infection. So something like HIV, perhaps you're a little bit familiar with it. It's something that is lifelong. It's something that stays in a person's body forever once they've gotten it. And that's because of this right here. It's because we have this reverse transcriptase that can take the RNA, turn it into DNA, and then that DNA, once it becomes double-stranded, can then put itself into our cellular genome. And then our cells will go through the regular process of making new cells and new cells and new cells. And as it's doing that, it's just making this over and over and over again, and it stays within our body forever. So the provirus stage is similar to the prophage stage in the bacterial infection during the lysogenic cycle. However, unlike the prophage, the provirus does not undergo excision. So remember when we looked at the bacteriophage, the prophage, once it gets into a situation where nutrients are depleted or the temperature has changed, things are a little bit uncomfortable in the bacterial cell, then that prophage is going to excise itself, cut itself out, loop itself, and then go through the process of making copies of itself and get out of that cell to go infect others before that cell dies because of whatever's going on. In the case of the provirus, that does not happen. It doesn't take itself out of the equation at any point in time. So we can see this in this illustration here. This is for the HIV virus. So you can see here, this is the HIV fuses to the host cell surface. So remember we have this attachment here. So we have the CD4 is this receptor here. And we have these on cells. You can see more of them here. These are CD4 cells that are on top of, or that are on the membrane of our T helper cells, or an immune system cell. So we have this CD4, and then you can see the spike here. This glycoprotein is going to attach to the CD4 here, and then this is going to allow this mechanism to occur, where we have this kind of bending down of the virus, pulling it down because it has attached to the CD4. It also uses this co-receptor, and then the two are going to join and pull the virus down toward the cell, toward the membrane of the cell. And in this case, you can see this is the fusion. This is not an example of endocytosis, where it actually engulfs the entire virus. This is an example of fusion because we see the envelope on the HIV virus, and that envelope is going to merge with or fuse with here the cell membrane. So in one, HIV fuses to the surface of the host cell, and then in two, HIV RNA, reverse transcriptase, integrase, because it's going to integrate itself, and other viral proteins are going to enter that host cell. So you can see it coming in here because that fusion has occurred. So it's going to get into the cell. Then our viral DNA is formed by reverse transcriptase. So you can see here they show this reverse transcriptase enzyme, the little dot there. You can see the viral RNA in here. And then reverse transcriptase is going along here, doing its thing, reverse transcribing so that it can make DNA. So it's making our single-strand DNA, and then it's going to make our double-strand DNA. So then when we have our double-strand DNA, our viral DNA is then transported across the nucleus. So this purple right here is showing the nuclear envelope. And the holes here are our nuclear pores. So our double-stranded DNA is going to make its way through one of these nuclear pores and get into the nucleus, which is within this purple region. We have our enzyme here, our integrase, which is going to go in. You can see it on both sides here. The integrase is going to then nicely merge the host DNA, the blue, and put that DNA from the virus into the host DNA. And at this point, when it has been integrated, it's called a provirus. So then if we scroll down here to see the rest of the process, now we have new viral RNA is used as a genomic RNA and to make viral proteins. So our body, this particular cell, will go along doing what it does. It's going to go through, it's going to transcribe mRNAs, just like it normally would, mRNAs, and those mRNAs are going to now contain the virus in it. So we're going to have RNA information that contains the virus, the information for the virus. Then those will leave the nucleus as RNAs do, and then it will go through the process of translation into proteins. These are supposed to be proteins here. And that's just the body doing what it does. It'll go through the process with the rough endoplasmic reticulum. It'll utilize ribosomes, the Golgi apparatus, all of these things to then make these viral proteins. Then the new viral RNA and proteins move to the cell surface, and a new immature HIV is going to form. And so all of these things are going to bud out of the cell. So we have all of our different pieces. They're not actually put together to make a mature virus at this point, but they are going to be contained within this envelope at this point because we've created an envelope using this budding out or exocytosis process. Then we have the virus maturing when protease releases the protein that forms the mature HIV. So then we have protease, which is another enzyme, is going to go in and it's going to take all of these different pieces and cleave some of them, which is to cut them and piece them back together and make them into our mature virion or virion. And you should know this process. This is an important process to understand and to know specific to HIV, but then also as a general process for viruses in human hosts.